Hello everyone, I am Dr. Katali and I will be your guide as we explore the fascinating world of biochemistry. Welcome to our lecture series. Today, we will embark on a journey to a very foundation of life itself. We're going to start an introduction to biochemistry and then zoom in on the fundamental unit of life, the cell. So, what exactly is biochemistry? As its core, biochemistry is the branch of science that explores the chemical and physicochemical processes and hello everyone. I am Dr. Katali and I'll be your guide as we explore the fascinating world of biochemistry. Welcome to our lecture series. So, today we are embarking on the journey to the very foundation of life itself. We are going to start with an introduction to biochemistry and then zoom in on the fundamental unit of life, the cell. So, what exactly is biochemistry? As its core, biochemistry is the branch of science that explores the chemical and physicochemical processes and substances that occur within the living organisms. It's the chemistry of life. We will be asking questions like what are we made of? How do our bodies get energy from food? How do our genes direct our cells? All of these answers lie in biochemistry. To understand the biochemistry, we first understand its stage and its actors. The stage is the cell. All organisms, from the simplest bacteria to complex human beings, are built from cells. All animal tissues, including those in your own body, are organized from collections of these fundamental units. The cell is, therefore, the fundamental unit of life. If a cell dies, the tissue, its parts of being begin to die, and that tissue can no longer function properly. It's that critical. The modern understanding of the cell, or the cell theory, rests on few key principles. Very first one is the cell make up all living matter. Number two, all cell all cells arise from other or pre-existing cells. Number three, genetic information, the blueprint of life, is passed from one generation of the cell to the next. And finally, all the chemical reactions of an organism, its metabolism, both the building up that we call anabolism and the breaking down, the catabolism, take place within the cells. So, in nature, we generally find two fundamental types of cells. Like the very first one is the prokaryotic cell, and the second one is the eukaryotic cell. As you can see on this slide, the eukaryotic cell is far more complex with its internal structures or organelles. These organelles are like specialized little organs within the cell. each with the specific job. So, before continuing to the individual organelles, let's differentiate between prokaryotic and eukaryotic cells. The slide highlights the key differences, which essentially boil down the complexity. The prokaryotes, like bacteria, are simple and smaller, and the size from 1 to 10 micrometers, while the eukaryotic cells are larger in size, and their size range from 10 to 100 micrometers. And furthermore, prokaryotic cells are mostly the unicellular cells, right? They are unicellular. What about eukaryotic cells? They are multicellular. They have complex structures or organelles. Number 3, prokaryotic membranes or prokaryotic cells got single membrane that is surrounded by rigid cell wall, while the eukaryotic cells got lipid bilayer structure with proteins in it. Furthermore, anaerobic or aerobic is the mode of production of energy, or the mode they use to produce energy, while the eukaryotes are aerobic in nature. So next one is the prokaryotic cells are not well-defined nuclear components, or not well-defined nucleus structure is there in their structures. Only a nuclear zone with the scattered DNA is there, while the eukaryotic cell got nucleus with well-defined 4 to 6 micrometer in diameter, contains DNA, and surrounded by a perinuclear membrane. Number 6, histones are absent. These are the specialized proteins. They are absent in prokaryotic cells, while eukaryotic cells got histones. Prokaryotic cells, there are no nuclei present, while eukaryotic cells got nucleolus, and they are rich in RNA. What about the prokaryotic cells? And the difference will continue with the point number 8. The prokaryotic cells, cytoplasm contain no cell organelles. What about the eukaryotic cells? They got well membrane bounded organelles. Prokaryotic cells present free in ribosomes are present freely in the cytoplasms, while in eukaryotic cells, ribosomes studded, or they are embedded on outer surface of the endoplasmic reticulum, that we call the rough endoplasmic reticulum. In prokaryotic cells, mitochondria is absent, while in eukaryotic cells, mitochondria is there, that is called the powerhouse of the cell, and enzymes of energy metabolisms are located inside the mitochondria. Next, we go to Golgi apparatus. That is absent in case of prokaryotic cells, while in eukaryotic cells, Golgi apparatus is present. Next is lysosomes. Lysosomes are absent in prokaryotic cells, while in eukaryotic cells, lysosomes are there. These are single membrane vesicle containing packets of hydrolytic enzymes. And what about the division of the cell, or the multiplication of the cell? In prokaryotic cells, cell division mostly follow by the fission reaction, the fission mode. What about the eukaryotic cells? They divide by mitosis, a programmed cell division process, right? What about next? Prokaryotic cells does not guard skeleton, while cytoskeleton is there in case of eukaryotic cells. RNA and protein synthesis in the same compartment in case of prokaryotic cells, while in eukaryotic cells, RNA is synthesized and processed in nucleus, while the protein synthesis occur in cytoplasm. And there, at the last, there are some of the examples of prokaryotic cells and eukaryotic cells. Bacteria, cyanobacteria, and Rickettsia. These are the examples of prokaryotic cells, while the fungi, plants, or animal cells, these are the examples of eukaryotic cells. So let's dive into these organelles. The eukaryotic cell contains many membrane-bound compartments that carry out specific cellular processes. The chief organelles we will discuss today are the nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, and finally the cytoskeleton. Nucleus. Let's start at the command center. It contains more than 95% of the cell's DNA and act as a control center of the cell. It's surrounded by a double membrane called nuclear envelope, which separates it from the rest of the cell fluid, the cytosol. Embedded in this envelope are nuclear pore complexes. These are not just holes, they are sophisticated gateways that carefully control the movement of molecules like proteins and RNA in and out of the nucleus. Inside the DNA, the DNA inside the DNA is coiled into a dense mass called chromatin, and a particularly dense region within the nucleus involved in making ribosomes is called the nucleolus. The fluid filling the nucleus, called the nucleoplasm, contains the enzyme needed for DNA replication and transcription. So, what does the nucleus do? Its primary function is to replicate DNA replication, making a copy of the genetic code before cell division, and RNA transcription. Transcription is the first step in reading the genetic information, where the specific gene's DNA sequence is copied into a messenger RNA molecule. This mRNA then carries the instructions out of the nucleus to build proteins. The next is the mitochondria. We have the powerhouse of the cell, the mitochondrion. The number, size, and even shape of the mitochondria can vary wildly depending on the cell's energy needs. A liver cell, which is very metabolically active, can have anywhere from 800 to 2500 of them. Its structure is key to its function. It's bounded by two very different concentric membranes. The outer membrane is relatively porous thanks to the proteins called porins that form channels. This allows small molecules to pass through freely. It also houses enzymes for things like fatty acid biosynthesis. The inner membrane, however, is a different story. It's highly selective and virtually impermeable to most substances. It's rich in proteins and unique phospholipid called cardiolipin. This membrane is folded into this membrane. The inner membrane is folded into these tight shelf-like structures called cristae, which dramatically increase its surface area. This is where the magic happens. The mitochondrion is a metabolic hub. It contains the enzyme for processing carbohydrates and fatty acids. But its main claim to fame is the rapid oxidation of molecules like NADH and FADH2, which are produced during early stages of metabolism. Through a process called oxidative phosphorylation, located right here on the inner membrane, the energy from this oxidation is trapped and stored in the highly energy bonds of ATP, the cell's main energy currency. Damage to mitochondria has critical clinical implications. For instance, it's a rare condition called Luft's disease. This energy conversion process is faulty. Furthermore, mitochondrial damage from free radicals is implicated in age-related disorders like Parkinson's disease and cardiomyopathy. Next one is the endoplasmic reticulum. Now, let's look at the cell's manufacturing and transport system. It's an extensive network of membrane-enclosed spaces that extends throughout the cytoplasm, from the nucleus all the way to the cell membrane. So, we got two different types of endoplasmic reticulum: the smooth endoplasmic reticulum and the rough endoplasmic reticulum. The rough endoplasmic reticulum is studded or embedded with the ribosomes, giving it rather granular appearance. Ribosomes are the factories where proteins are built and is to synthesize proteins that are destined for secretion, for incorporation into the cell membrane, or for other organelles. When we break open cells in the lab to study them, the rough endoplasmic reticulum fragments into small vesicles we call microsomes. What about the smooth endoplasmic reticulum? Smooth endoplasmic reticulum lacks ribosomes. Its functions are different. It's heavily involved in lipid synthesis, detoxification of drugs and poisons, and the modification of transport of the proteins made in the rough endoplasmic reticulum. Next is the Golgi complex. Think of the Golgi apparatus or the Golgi complex as the cell's post office and the packaging center. It is stacked up flattened, fluid-filled sacs called cisternae. It receives newly synthesized proteins from the endoplasmic reticulum in little transport vesicles. The Golgi complex then acts as a sorting device. It modifies these proteins, often adding sugar molecules to make glycoproteins or lipids to make lipoproteins. It then packages them and sends them off to their final destinations in new vesicles. Proteins without the proper address labels and or signal peptides are rejected and remain in the cytoplasms. Next is the lysosome. Lysosomes are essentially membrane-bound bags filled with powerhouse or powerful digestive enzymes. They maintain an acidic internal environment with a pH around 5, which is perfect for the 30 to 40 different hydrolytic enzymes they contain. These enzymes can break down almost any biological polymer like proteins, nucleic acids, lipids, and the carbohydrates. As long as the lysosomal membrane is intact, these enzymes are safely contained. But if the membrane ruptures, the enzymes are released and can digest the cell itself. This brings us to their biomedical importance. Lysosomes are crucial for autophagy. Like the autophagy, the cell's way of cleaning up its own damaged components, like the old mitochondrion. Defense, white blood cells engulf bacteria and then fuse with lysosomes to digest them. Post-mortem autolysis. After death, lysosomes break down and their enzymes cause self-digestion of the tissues. Fertilization. The acrosome of the head of the sperm cells is specialized lysosomes that help it to penetrate the ovum or the egg. Furthermore, a whole class of inherited disorders called lysosomal storage diseases are caused by the absence of a specific lysosomal enzyme. Without it, substances like complex lipids or polysaccharides accumulate inside the cells, leading to a very severe medical problems. Next is the peroxisomes. Let's briefly touch on peroxisomes. These are the small organelles called microbodies. Those are involved in oxidation reactions. They are particularly important for breaking down very long-chain fatty acids. A key feature is that one of their reactions produces hydrogen peroxide, which is toxic to the cells. But peroxisomes also contain enzymes called catalase, which immediately break that toxic hydrogen peroxide down into harmless water and oxygen. In the inherited disorder, Zolger syndrome, the peroxisomes are absent, leading to severe metabolic issues. Finally, we got cytoskeleton. That is a cell scaffolding and highway system. It's a complex network of protein filaments that gives the cell its shape. Provides mechanical support, and enables movement. It is made up of three main components: microtubules, microfilaments, and microtrabeculi. So, very first one is microtubules. These are long, hollow cylindrical structures that are made up of protein called tubulin. They act like railroad tracks for moving vesicles and organelles within the cell, and they form the spindle fiber that separates chromosomes during cell division. Next one is the microfilament. They are long, hollow cylinders made up of a protein called actin. They are crucial for cell movement, muscle contraction, and dividing the cell into two. The third one is microtrabeculi. They are more delicate, transient network that may help organize the cytosol and its enzymes. Next one is the cytoplasm, the cytosol. We cannot forget the cytosol. This is the fluid or sap in which all the organelles are suspended. It's not just empty space. It's a highly concentrated solution of enzymes where many essential metabolic reactions like glycolysis take place. It also contains free ribosomes and supplies the energy and cofactors needed for protein synthesis on the rough endoplasmic reticulum. And that brings us to the end of our introductory tour of the cell and its organelles. We have seen how this intricate and highly organized structure is the stage upon which the drama of the biochemistry unfolds. I hope this has provided a solid foundation for our future discussions. Thank you for the attention, and I'm now happy to take any questions you may have in your comment section. Thank you.